Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about bioenergetics, how energy flows through the body. To understand bioenergetics, we're going to look at the basic chemical reactions that underlie metabolism. These are the four chemical reactions that underlie metabolism. We're going to talk about each. The first chemical reaction is catabolic reactions, or the catabolic pathway. Catabolic pathway will release energy by breaking complex molecules into simpler molecules. For example, you can take a molecule like a starch, which is composed of many glucose molecules. You can convert it into individual monomers called glucose, and you will therefore release energy. That is referred to as the exergonic reaction. The opposite is anabolic pathway, or anabolism. Anabolism is consuming energy to build a more complex molecule, such as taking amino acids, which are monomers, joining them together to form a polypeptide, which will eventually be a protein. This will require energy. This will absorb energy, so we call it an endergonic reaction. The next reaction we'll look at are called redox reactions. So redox reaction require the donation of electrons and the receiving of electrons. So oxidation and reduction reactions occur when electrons are transferred between donor molecules and acceptor molecules. A donor molecule is going to give electrons and become oxidized. An acceptor receives electrons and become reduced. These reactions always occur simultaneously. So in this image below, I have a redox reaction that is occurring. Glyceraldehyde 3-phosphate is going to become oxidized. It's going to give away an electron to NAD. NAD is an acceptor. It will receive the electrons and become reduced, therefore forming NADH. The product of glyceraldehyde 3-phosphate being converted is 1,3-bisphosphoglycerate. This is a redox reaction, and it must occur simultaneously. This is another way of viewing that same process, a redox reaction. Glyceraldehyde 3-phosphate is going to be converted into 1,3-bisphosphoglycerate. NAD will come and be an acceptor and receive electrons from G3P. NAD is the oxidizing agent. It will become reduced, meaning it will gain electrons. Three important electron carriers are involved in metabolism. We're going to talk about these three. NAD and FAD are seen in the mitochondria, whereas NADP will be seen in chloroplast. NAD is based on niacin, which is a vitamin B3 derivative, and FAD is derived from riboflavin, which is a vitamin B12 derivative. ATP production and energy storage is when organisms release energy from nutrients in the form of ATP. In this image, we see that ATP has 3 phosphate groups. If we reduce it by removing a phosphate group, it becomes ADP. Now it has 2 phosphate groups. We can further reduce it to AMP, which would only have 1 phosphate group. The energy is stored in these covalent bonds that we're going to call high energy bonds. So the more phosphate groups this molecule has, the more energy it will possess. Enzymes phosphorylate ADP into ATP in 3 ways. One form is called substrate-level phosphorylation. The second one is oxidative phosphorylation. And the third one is photophosphorylation. The first way to generate ATP is called substrate-level phosphorylation. This occurs when an organic phosphate is added from a substrate directly to ADP. So whenever we say I'm making energy, you are creating ATP, a molecule that has 3 phosphate groups. So if you already have ADP present, a substrate can contain a phosphate group. It can give the phosphate group to ADP, therefore creating energy. So that is called substrate-level phosphorylation. In this image I created, we have 1,3-bisphosphoglycerate. It has a phosphate group on carbon 1 and it has a phosphate group on carbon 3. This is a substrate. ADP is going to receive one of those phosphate groups. On carbon 1 of 1,3-bisphosphoglycerate, it will donate the phosphate group to ADP, therefore creating energy. This is called substrate-level phosphorylation. The byproduct is 3-phosphoglycerate. The second way we can make energy is by losing electrons to the intermembrane space. When these coenzymes, NAD and FAD, receive their hydrogen, they're going to take their hydrogen or electrons to the mitochondria. They will donate the electrons to the intermembrane of the mitochondria. Electrons will move, and therefore the intermembrane of the mitochondria will pump hydrogen into the intermembrane space. This is going to create a concentration gradient. Then these electrons are eventually going to flow through a enzyme complex called ATP synthase complex. That complex is going to generate ATP. This resembles the turbine in the dam. The water concentration behind the dam is potential energy. When water flows through the dam, through the turbine, it spins the turbine. The turbine is going to convert potential energy into kinetic energy. If you think of that scenario, that's what's happening in the mitochondria. On the outside of the intermembrane of the mitochondria, you have the intermembrane space. This is where the high concentration of hydrogen is going to be created. That is the water behind the dam, or the hydrogen behind the intermembrane. When this hydrogen flows through an opening that contains ATP synthase complex, it's going to spin the turbine, therefore generating ATP. So this is the second way to make energy. The third way to make energy is through light. So the use of light energy will generate energy, and it is called photophosphorylation. We will cover this extensively when we talk about plants.